All right, folks, I'm going to get started. So I want to give a really high-level intro for this session just to set the stage. And that is, if you've listened to the first intro recording from today, this slide is familiar to you, is just briefly explain what is the difference of using PacBio long reads for single-cell sequencing. And I hope this graphic really demonstrates that when you use short reads for single cell, you're really only capturing the gene level information due to the limitation of the read lengths. And this is because the single cell tag information, such as cell barcodes and UMIs, are only at the ends of the molecule. Therefore, when you have short reads that cannot sequence the entire molecule in one go, you can only capture the gene ends. In contrast, with PacBio single-cell Iso-Seq, you can get full-length isoforms with high accuracy, and you can capture the UMI and barcode information. In the next talk, as well as the next session, the last live session of today, you will hear both bioinformatics and also analysis of what's called the Moss-seq or Moss-Iso-Seq method. And I just want to briefly give you a background for this. What Moss-Iso-Seq is based on the preprint that is shown here by authors at the Broad Institute. It is a general amplicon concatenation method that, as the first kind of use, we're applying it to single cell cDNA, and particularly for the 10x platforms. And the reason we're doing this concatenation is that the PacBio hi-fi reads have a really long read length. Typically, a whole genome sequencing library would have somewhere between 15 to 16 kilobases. But we know, for example, the cDNA generated from the 10x systems are generally around 600 to 1 kilobases. Therefore, applying the Moss-seq method, we can actually create a much larger insert size that can still obtain high-quality reads on the PacBio systems and increase throughput in this manner. What the Broad authors demonstrated was that with a single SMRT cell, with the Moss-seq method, they can achieve 33 million molecules. They show that with 33 million molecules on a single SMRT cell, they can recapitulate the cell types that they're seeing in the matching short read data. But they also get something that you can't see with short read data, which is alternative splicing events. That's shown here in the lower right corner for the CD45 gene. Here, what they're showing is different isoforms, such as RB, RAB, and using the color codes, they show that different isoforms are found in different cell types. In the next talk by Roger, you'll hear more about details of the SMRTLink analysis for single-cell Iso-Seq that we're about to release at the end of this year. In conjunction, this is done with the release of our Moss-seq kit, which is a commercialized version of the preprint method that you've seen in the previous slide. This kit will support and is intended for use with 10x Chromium single-cell 3' cDNA. It will take about 2 days to do the library preparation and about 32 to 34 hours for sequencing. More details of the kit will be available when we release it at ASSG. And then for the informatics, you will hear it from Roger today. And with that, Roger, can you... All right, cool, take it away.
Thank you, Liz, for the intro. I'm Roger. I'm a bioinformatics scientist at PacBio, and I'm going to be talking about the single-cell Iso-Seq workflow for the bioinformatics for this Moss-seq kit. First, I want to motivate this. I want to ask, how can we go from hi-fi reads to an isoform level cell type cluster? And the answer, as Liz just gave us, is through concatenation to be able to increase our throughput by over an order of magnitude. Now, what this will look like for customers is a push-button workflow through SMRTLink. And when you get the SMRTLink 11.1 update, you'll be able to do a read segmentation and a single-cell Iso-Seq workflow. And this will provide various statistics regarding your read segmentation. So it'll tell you how many hi-fi reads and how many segmented reads that you get out of this, as well as the percentage of your ZMWs that have full-length arrays and your mean array size, as well as QC plots like this concatenation factor histogram, where it'll show you the percentage in each bin. We'll also provide S-read statistics. This was just the number of reads that you get at each step in your processing. So it'll tell you how many reads have valid barcodes, how many after get corrected, and how many reads you have after deduplication of your UMIs. Another QC plot that we find pretty helpful is hi-fi read length distribution by the concatenation factor. So each of the bins in this hi-fi read length distribution are colored by the concatenation factor of that molecule. In addition, we'll also tell you some statistics about how many cells that we think are in your sample, as well as the percentage of reads that are inside what we consider to be real cells, the mean reads per cell and the median UMIs per cell. We will also provide transcript summaries, and this will be like a Sconti-3 replacement, where we'll tell you exactly the structural categories and the breakdown of each of these for all of your transcripts. To get more into the details of the exact tools that are getting used within this pipeline, this is really getting separated into two workflows in SMRTLink, that being the read segmentation and the single-cell Iso-Seq pipeline. Now, the read segmentation only has one tool, and this is what we call Scira. This is our read segmenter. For the single-cell Iso-Seq pipeline, there are three main steps, each having their associated tools. So for the primary UMI barcode extraction, we have lima, tag, and refine. For correction and UMI deduplication, we have, oh, I skipped a slide, that's fine. Correct and group dedupe. And for the mapping and transcript classification, we have PBMM2, collapse, and pigeon. So this is, I'm going to be going over each of the tools and how to run them in the following slides. Also, I should point out, because I skipped this, the output from Pigeon is going to be compatible with tertiary software analysis packages like Syrah, ScanPy, and Kana. So this will enable you to do all of your cell type clustering. Okay. The first tool after you have your hi-fi reads from CCS is Scira. And what Scira does is it will deconcatenate your reads into its constituent cDNA molecules. So here, let me laser pointer, we have a concatenated molecule with labeled adapters A through E with 4 constituent cDNA molecules in between. After Scira, what this will do is it will excise all of these adapters and only retain the cDNA molecules that are bound by 2 adapters. So this will result in 4 cDNA molecules. And by making arrays with 16 cDNA molecules, we can have an increase in our throughput by 16x. The command for this is actually pretty simple. It's just a Scira split, the number of threads that you want, your hi-fi reads, your adapters, and then your output. Following this is what our molecule topology looks like, where we have the 10x TSO, our cDNA, poly-A UMI cell barcode, and the 10x 3' primer. Using lima, we're able to extract the 10x sequences, so we're just left with a cDNA molecule with the barcodes attached to it. And for this, you'd want to use the Iso-Seq preset on lima. After this, what we want to do is extract the UMI and cell barcodes from each of these transcripts. And when we do this, it will get excised from the sequence in the BAM entry, and afterwards it will be shunted into BAM tags, being XM for the UMI and XC for the cell barcode. When you're actually running this command in the command line, you need to specify a design for your transcript. And for the 3' version 3 kit of for the 10x chromium, the design is transcript with a 12 base pair UMI and a 16 base pair barcode. And again, this is pretty simple. It just takes your lima output and outputs your tagged BAM file. If you were to homebrew this, you would have to change the transcript design, and that is technically supported through SMRTLink in the advanced options. After this, we go through Iso-Seq 3 refine, and all this does is it will check for a poly A or a poly T signal in your sequences, and it will also check for artificial concatenators based off of the 10x sequences that you also give it. So if there's like a PCR chimera, it should be able to get rid of those if there's a 10x primer in the middle. And again, all this does is it won't affect your cell barcode and UMI BAM tags. It will just excise the poly A or poly T signal. And yeah, the parameter that we use to run this is require poly A. Following Iso-Seq 3 refine, we had to make a new tool for barcode correction, and this is Iso-Seq 3 correct. So this will take a list of canonical 10x barcodes, which you can get from Cell Ranger and Space Ranger, and it will take your CDA sequence and your tagged barcodes, one of which, for demonstration purposes, I put a mismatch in the cell barcode. And after we run correct, what it'll do is it will correct this cell barcode with a mismatch to a canonical barcode that's in your include list here. And after that, the BAM tag will change from XD to CB, and it will no longer have that mismatch.
2 minutes, Roger.
2 minutes?
Yeah.
Yeah, for this, we need to reverse complement the barcode list. And then after this, we sort with SAM tools, excuse me. And we're sorting by the corrected barcode tag. Following this, we do group dedupe, which will essentially take your molecules and look at their UMIs, and if they match, it will collapse those into one molecule and produce counts, which also get put into BAM tags. Following this, once we have deduplicated molecules, we will align them to HG38, and this will produce your splice alignments. And this is using the Iso-Seq preset, and we also like to sort the output. Okay. Once we have our spliced alignments, what we can do is we can look at these and look at redundant isoforms within our data set and collapse them using Iso-Seq 3 collapse into unique isoforms. And after this, we will need to sort the GFF output for use in Pigeon. What Pigeon does is this is a drop-in replacement for Scanti, and it will classify your isoforms into their own structural categories based off of a reference transcriptome. So it will take your alignments, or your unique isoforms, I should say, and it will label them with their structural categories. And additional parameters that you'd want to specify are a list of poly A sequences, as well as cage peak information to tell you whether or not your ends are actually real. And this will produce a tab delimited classification file. Thereafter, we can run Pigeon filter, and this is the same as Scanti filtering, where we'll take out like genic introns, genic genomic isoforms, or remove things that were a product of premature RT switching or interpriming. And this will give you a nice clean set of isoforms, which we can then run Mixera on to make our 3 output files, which will be compatible with Kana, Syrah, and ScanPy. And this will give you your nice cell type clusters based off of your isoform level information. Sorry, I kind of blazed through the end kind of fast, but yeah, that's it. I'd like to thank the wonderful team at PacBio and the guys over at the Broad Institute that were instrumental in making this happen, as well as Scott from the Fred Hutch. And, yeah.
Thank you, Roger. There were some questions. I did answer some of them, but feel free to check them out during Anoushka's talk. In the interest of time, I'm going to bring up Anoushka's talk. And Alex as well. All right, take it away.
Okay, excellent. Thanks, guys, for having me over. Today, I'm going to be talking about visualization tools that we and other people have built. And this is work that was led by Alex in the lab. He was a research tech. He's here to answer all of the technical questions today. And with that, well, I think Roger went over most of the single-cell Iso-Seq, so I don't need to go into the details. I do want to point out that splicing expands the proteome, which is why we really care about it from a biological perspective. And just like one of my favorite facts is that the worm, mice, and humans, we all have the same number of genes, but we all have very distinct number of isoforms. And these isoforms manifest themselves in a tissue-specific manner, in a region-specific manner, and a cell type-specific manner. And I guess you guys are very, very veteran users of PacBio, so I don't think I need to give you details, but let's quickly go over how splicing is measured and how we visualize it. So if you have a hypothetical gene with two alternative exons, they can lead to 4 distinct transcripts, right? Where you have both exons included, one exon included, or neither. And in a sample, what that would look like is, for example, you have these 4 isoforms being expressed in these like 5 reads for the first and 1 read for the last, etc. What that traditionally was measured as was like a PSI count, where you count the number of reads with a particular exon over the total number of reads. So in this case, both the red and the blue exons have a 66% inclusion. However, and this was like, I'm guessing most of you recognize a sashimi plot, where you have a pileup of the different number of reads, and you can see that this cassette exon was lowly included in this red cell type endothelial, and mesenchymal cells have a lot of it. The problem with this approach is that if you have like 2 different samples with the same PSI value, these can look very, very different on the like expression level. And there's no connectivity info if you look at the single exons. So what we did instead was looked at the percent isoform. So you just look at the number of reads per isoform over the total number of reads, and you can get PI values for them. We implemented the Scissor-seq or single-cell Iso-Seq workflow a few years ago, wherein, well, again, Liz mentioned what that is, so I won't go over it. And then we also implemented an R package, which takes you right from the FASTQ files, barcode deconvolution, all the way down to visualization. The visualization that we spoke about looked at between sample comparisons on a genome-wide level. So this is just a quick summary of what we did. If you look at the prefrontal cortex versus the hippocampus, and you're trying to measure differential isoform expression for the same cell type across 2 different brain regions, or for different cell types within the same brain region, what we did was we showed the number overall percentage of the genes that were tested for differential isoform expression, and how many isoforms, for example, contributed for there to be significant differences between the 2 samples. So this is kind of a global level approach in terms of visualization. And same for if you're doing multiple samples, you can basically build a heat map where each tile here represents, say, between the vascular cells and the excitatory neurons, how many genes are differentially expressed. This is really good for like a bird's eye view of what differential isoform expression looks like. If you want to zoom in on specific genes, what we did was a similar heat map where you have different cell types as the columns and the isoforms that we care about as rows. The problem with this, it's not really a problem, it's just that I was painstakingly making all of these boxes, which is bioinformatically not a smart thing to do. And so we wanted to come up with a better way of representing this. We did the same thing with spatial isoform sequencing. So it's the same principle where you sequence long reads and short reads from the same sample. In this case, they're spatial barcodes. And you can really visualize how the different isoforms are expressed in, say, the front of the brain versus the back of the brain for given genes. Other labs and other methods have come up with different visualization tools. This is one from the Mortazavi lab at UC Irvine, where they came up with this really pretty way of showing the different isoforms that are expressed. And you can sort of see that in a similar heat map fashion with the different isoforms. And here it shows the degree to which that isoform is expressed. GG transcript is a new tool wherein you can see the individual transcripts, and you can show either by the weights of the line, thickness of the line, or percentages how much each isoform is expressed. However, I'd like to say that both like the approaches I just showed you by other labs as well as ours leave something to be desired. There's a few things that are missing. So what we want to see is a way to visualize a large diversity of cell types. And instead of having to like squint to figure out which the different isoforms are there, we want to see it all at once. We want it to be easy to implement, no manual drawing of isoforms. And we want there to be customizable parameters with the ability to add in like sequence elements, for example, SNPs or RNA modifications, or any sequence elements that we really want to visualize. So enter Scissor-wiz. So this is the package that it's an R package that Alex wrote, and we've been using it in our publication sort of informally for a while, but now he's made it really, really easy to implement for any gene. So I'm just going to show you what the output looks like. On the bottom, for every plot for every gene, what we see is the gen code annotation. So if you want to see what isoforms for a given gene, something as complex as CAMK2B, which is a synaptic gene, you can see those on the bottom. Piled up on top is going to be different isoforms for any cell type that you choose. So in this case, I'm showing granular neuroblasts, and you can see that there are like the exons that pile up that are constitutively expressed are in one color, and the ones that are alternative are in a different color. So you have orange here, and the red exon here is actually not an exon, it's an extension. And so that is something that can be customized if you go into the code and just like add in a couple parameters. So here you can see for this particular example, you have one isoform that has this red exon. You can see the same, for a different isoform, which has only this orange isoform exon, or so on and so forth, where you have this orange isoform or both of them together. You can do the same, pile up the reads for different cell types, and here you can see that the isoform patterns are changing. And here I have excitatory neurons, inhibitory neurons, and granular neuroblasts, and you can really see how the different cell types modulate the use of these three different exons, and therefore like 4 or 5 different isoforms, depending on whether these are included or excluded together or separately. So it really provides this comprehensive view of the entire isoform repertoire in a sample. And you can easily imagine here I have the 2 inhibitor excitatory neuron cell subtypes as being separate, but you can group them together. And there is no reason for this to be only single cell isoform sequencing. You can have a case and control sample. You can have 2 different bulk long read sequencing experiments. And all you need to do is tell the program that cell type 1 or group 1 is, Alzheimer's disease, and group 2 is the control samples. The requirements for Scissor-wiz are very, very basic. You need an R installation. We're working with version 3.5 or above. You need a gen code or any type of annotation. We need the reads to be in a GFF format, so we know exactly what the exon coordinates are. And then you need a gene association, so we can like exactly trace back the gene of interest. And lastly, I care more about the pretty colors, but you need a cell type file or, some sort of case control identifier so that it will pull them out of your reads. So the workflow for Scissor-wiz is you start off with you download the Scissor-wiz and install the package. You create your cell type file. Then you have 2 options. I'm sorry, these boxes got a little bit cut off, but you have 2 options. You can either use the Scissor-seeker implementation, the one I was mentioning before, where you can totally just plug in a button and the file is ready for you, or you can come in with your own set of files. I'm so sorry, I don't know why this is cutting off the boxes, but basically you need a read to gene mapping and a GFF file. And then lastly, if you want to visualize your RNA modifications or SNPs, you can run a mismatch finder, and you can also implement that as an optional step. So this is kind of the whole workflow. Again, boxes are being weird. And for the mismatches, what you can do is what you end up seeing is different colors for either insertions, deletions, or substitutions. And if you're working with really fun RNA modifications or SNPs for, say, Alzheimer's disease, you'll be able to see them very, very easily in your different samples. So with that, I'm going to just give you a few conclusions. Scissor-wiz can be used on any gene, and it's really helpful to point out the novel isoforms, given you have the gen code annotation on the bottom. It integrates seamlessly with Scissor-Seeker, and we recommend it, but you can totally come in with your own files with the recommended format. You can use this for case control studies or any like bulk sequencing studies as well, as long as you have that identifier in the read name. One thing I didn't mention was that you can cluster on either the transcription start site or the end site. So if there's differences on your TSS usage, you can really visualize them in the plots as well. Any number of cell types can be plotted. We also implemented a zoom functionality if you want to explore very complex looking genes. And the alternative exon PSI cutoff can be set. So if you only want to visualize exons that are massively alternative and not like, 95% inclusion, you can change that. And colors, that's a me thing, but they can be changed, and that makes me very happy. With that, I'm happy to take any questions. Please check out Alex's publication that came out in Bioinformatics earlier this year, and the GitHub link I can also drop in the chat that has more documentation and the exact parameters you can include when you are trying to run the tool. Thanks.
Thank you, Anoushka. There is one great question from Antonio. So does Scissor-Seq get you the PSI of the exons between the conditions, like between cell clusters or samples?
No, it sort of calculates that within Scissor-wiz. It calculates it within the running of the files themselves, and what you get out is just a visualization. I'm sure if that is something you wanted, you could totally print it out either in the code, or we can add it for you if that is going to be helpful.
Awesome. Well, thank you. We have one last session, so I'm hoping to see all of you at the live session in a minute. See you later.
Thanks. Bye.